Hello everyone, my name is Leilani Lucas and I'm a professor of anthropology at the College of Southern Nevada. My talk today aims to demonstrate archaeology in action by showing how archaeobotanists reconstruct ancient culinary practices through the analysis of charred food remains. First, thank you to Mandy for inviting me to share my research. So over the past decade, advances in the analysis of charred food remains have opened new ways to investigate past food production. For me, the key point is to move beyond macrobotanical remains alone and rather to bring together plant remains, cooking technologies, experimental archaeology, and ethnographic comparison. I believe this is especially important in places like Cyprus, where preservation, recovery histories, and older research priorities have left what we can see about everyday food practices limited. In this talk, I will use Bronze Age Kissonerga-Skalia as a case study to explore how these different lines of evidence can help us think about the evolution of culinary traditions in prehistoric Southwest Cyprus, and how small, charred fragments can connect to much broader questions about food, cooking technologies, and tradition.
So to understand what I am investigating, we need to start with cooking itself. It has been argued that cooking helped make us human. Wrangham's cooking hypothesis, which essentially links cooking to biological changes such as smaller guts and larger brains, remains debated. But what it does is highlights an important point. Cooking is never only biological. For humans, cooking is also cultural. It changes raw ingredients or foodstuffs into actual food through learned practices, shared knowledge, and social expectations. The sharing of meals and the development of culinary practices are central to how communities form, remember, and reproduce identity over time. So when I talk about prehistoric charred food remains today, I'm not only asking what people ate, I'm asking how food was prepared, transformed, shared, and made meaningful. Food, then, is never simply about nutrition or calories. Culinary practices are embedded in cultural traditions, social organization, labor practices, and even shared knowledge systems. The way in which people prepare and consume food are learned, repeated, and transmitted across generations. Because of this, food preparation can give us insight into identity, social relationships, and daily life in the past. The challenge is that these practices are often difficult to identify archaeologically. Archaeobotanical research has traditionally focused on identifying plant taxa and reconstructing agriculture or subsistence practices. Those questions are important, of course, but they do not always tell us how plants were transformed into food. This is where charred food remains become especially valuable. Archaeologists reconstruct ancient foodways through multiple lines of evidence. These include plant remains, grinding tools, cooking installations, ceramic assemblages, residue analysis, and many other forms of material culture associated with food preparation and consumption. Each line of evidence tells us something different about how food was produced, how it was processed, prepared, and consumed. Charred food remains are especially useful because they sometimes preserve traces of preparation themselves, including how the food was processed, how it was mixed together with other ingredients, how it was heated, and eventually cooked. Yet these remains have often been understudied or classified broadly as unidentifiable. However, with newer methodological approaches, this is beginning to change. To address this intersection between culinary tradition and cuisine, I will frame it based on Fuller and Champion's 2023 schematic representation of post-harvest processing. This is essentially the stages between collection of a natural foodstuff, which is labeled 1, and its conversion to a cultural food, which is to the far right. This is distinct from the mode of subsistence, so how the food was acquired, whether it was hunted or foraged, is not taken into consideration in discussions of these types of labor intensifications. This is not to say that acquisition does not play into post-harvest processing, but rather today I'm going to focus on the latter. So you might ask yourself, why should we care about labor intensification post-harvest? I believe this is an extremely valuable way to understand the evolution of culinary traditions and foodways, and further, it possibly explains the diversity of crop or ingredient choices and the evolution of cooking technologies or equipment. Fuller and Champion's schematic outlines 4 stages of labor intensification and processing that occur before cooking, including 2 potential aspects of intensification. We have labor processing 2 and the materialesque intensification labeled 4. They argue that the total degree of post-harvest intensification in the creation of a culturally edible food at the end is the sum of numbers 2 and 4. I have added another stage, 5, for artifacts that are also created before cooking, but rather than being used for the cooking process, like cooking equipment labeled in 4, the artifacts are used for consumption and are performed after cooking. So I believe the artifacts of consumption also tie into this kind of web of connections, driving cultural preference and change in culinary traditions, creating unique, regionally specific foodways. Although I will not be discussing stage 5 in further detail today, I wanted to say a little something about it. So it's all about artifacts of consumption. So this is what happens after cooking and/or preparation. These are objects used for serving and eating that reflect cultural preferences and social change. In Cyprus, these range from simple ladles and drinking cups to imported feasting artifacts. The labor investment goes from minimal intensification with simple functional tools like ladles to high intensification with large specialized vessels used for communal feasting and social display. I would also include terracotta representations of food, feasting, and dining scenes from the island, like the early Bronze Age Bunus bowl. These types of artifacts are high investment, but are less about functionality, but shape and reinforce culinary traditions in the past, linking food, technology, and social identity. By studying these objects as well, alongside the archaeobotanical data and material culture, I believe we can understand food culture in new ways. I'm using foodways as the broad umbrella term for a group's food culture, from acquisition and processing through cooking, consumption, and disposal. Within that broader system, culinary tradition refers to the learned practices and technologies that transform these ingredients into recognizable foods. Cuisine is the outcome of all of these practices, food as prepared, served, and understood within a particular cultural context. So these distinctions are useful because they help us move from plant remains as possible ingredients to food as a cultural product. One of the challenges in studying foodways archaeologically is that food preparation is often treated as ephemeral, something that happens and then for the most part disappears. But culinary practices can leave material traces. They appear in artifacts, installations, spatial organization, and in some cases, the food itself when it becomes charred and preserved archaeologically. The image on the left reminds us that food preparation was a socially visible and culturally meaningful activity, not just a biological necessity. Cooking activities leave traces in material culture. The image on the right shows the oven feature for Kissonerga-Skalia, and installations like this provide the context for asking how ingredients were transformed into prepared foods. So as in archaeology more broadly, context is everything. This is where charred food remains become important. They allow us to ask not only what ingredients were present, but what people were doing with those ingredients. But before moving on to the Cyprus case study, I want to introduce the main category of the material I will be discussing. And these are those amorphous charred objects, or the ACOs. ACOs are frequently recovered in archaeobotanical assemblages, but they are often recorded very broadly as unidentifiable remains. The problem is that this category can hide a lot of information. These fragments may look unpromising at first, but under low-power microscopy and further SEM, they can preserve traces of ingredients, processing, preparation, and cooking. Rather than treating ACOs as analytical leftovers, we can approach them as material traces of culinary practice, evidence of that transformation from ingredients into food. Over the past decade, microstructural analysis has shown that ACOs are not simply unidentifiable fragments. In a range of archaeological contexts, they have been interpreted to be everything from bread-like products to cereal preparations, porridges, mixed plant foods, or a wide range of other processed foods. What makes this possible is the preserved internal structure of the charred material. Under magnification, we can sometimes identify cereal tissues, bran, starch-rich matrices, seed fragments, fruit tissues, pulses, and other inclusions. These traces can tell us not only which plants were present, but how they were processed, how they were mixed and heated. That is the methodological shift I'm building from. Amorphous fragments are not necessarily at the end of the identification process. They can be the starting point for reconstructing culinary practice. To interpret these fragments more carefully, we need comparative frameworks. Recent work by Valamotti and colleagues, along with other studies combining experimental archaeology, ethnography, and microscopy, have been especially important here. Experimental work allows us to produce food remains under known conditions. Ethnoarchaeological observations help us think about real preparation practices, cooking equipment, and the kinds of traces those practices may leave behind. These comparative materials can then be examined using SEM and used as reference datasets. So the goal is not to make a simple one-to-one match, but to identify recurring microstructural features that help us interpret archaeological food remains more confidently. One of the key developments in this field has been the creation of experimental and archaeological reference datasets. Recent work at Çatalhöyük in particular is especially useful because they compared archaeological food remains with experimentally charred doughs, flatbreads, and porridges. Their work shows that amorphous charred material is not simply unidentifiable. Under SEM, different food preparations can preserve distinct microstructural features, including differences in matrix texture, porosity, cereal tissue, and the organization of the plant fragments. I am not using Neolithic Çatalhöyük here as a direct cultural comparison for Bronze Age Cyprus, but I am using it as a methodological example. That said, questions about oven-based cereal traditions moving from Southwest Asia to Cyprus are part of the broader background to my research. For today, the important point is that the reference material helps us move from charred remains to more careful interpretations about possible food preparation in the past. If Cyprus followed a different trajectory from the mainland, did food culture diverge as well? This question came out of my earlier work on Neolithic and Chalcolithic Cyprus, where the island appears to follow a somewhat different path for the mainland in terms of subsistence choices. But the question does not stop in the Neolithic. It becomes especially important in the Bronze Age, when communities in southwest Cyprus were increasingly connected to wider Eastern Mediterranean networks while still maintaining local traditions. So when we get to Kissonerga-Skalia, the question becomes more specific. Were new ingredients and technologies simply added into everyday practices, or were they transformed into something locally meaningful, something possibly distinct to prehistoric Cyprus? So why the island of Cyprus? Well, islands provide interesting case studies because they often magnify evolutionary and cultural changes, making processes more visible. In biology, island biogeography can explain why species adapt rapidly, as seen in the dwarfism of Cyprus's pygmy hippopotamus.
In terms of human occupation on the island, it was settled late, despite its proximity to mainland Southwest Asia. However, once occupied, settlement patterns tend to be shaped by Cyprus's arid Mediterranean climate with short, wet winters and long, dry summers. Rainfall is highly variable with recurrent droughts impacting agriculture and water availability. Early sites mostly clustered near river drainages, but by the Bronze Age, communities shifted to less fertile areas, coinciding with the reintroduction of cattle and the introduction of plow technology. The island is known for two resources primarily, picrolite and copper, a legacy still reflected today in its euro coin, which features a Chalcolithic picrolite figurine. And beyond material prehistory, Cyprus is also rich in mythology, famously known as the birthplace of Aphrodite, linking the island's past to both its archaeological and cultural heritage. For additional context, human occupation on the island reflects a complex history of migration and adaptation, with multiple episodes of plant and animal translocations from the mainland Levant. Initial exploration during the Epipaleolithic, the Akrotiri phase, may have contributed to the extinction of the endemic pygmy hippopotamus. Later, the Cypriot pre-pottery Neolithic A saw the arrival of hunter-cultivators, followed by Cypriot early pre-pottery Neolithic B farming populations who introduced plant and animal species from mainland Southwest Asia. At this stage, Cyprus closely resembled the mainland in material culture, architecture, and small-scale intensive agriculture. However, over time, subsistence strategies and material culture diverged. Unlike the mainland, which became increasingly dependent on cereal agriculture, Cyprus seemed to retain a mixed subsistence economy with hunting of fallow deer an important part. By the late Chalcolithic, conflicts between deer hunting and cereal cultivation prompted efforts to reschedule agricultural labor, shifting harvests later in the season. A growing reliance on cash crops and caprine husbandry possibly suggests adaptation to social and economic pressures, including early signs of urbanization and expanding trade networks. In the Bronze Age, copper production became a driving force, shifting settlements to less fertile areas. This transition necessitated plow-based agriculture and extensification and greater integration of livestock, ultimately increasing agricultural reliance. The shift wasn't inevitable, but likely a dynamic response to environmental, social, and economic factors. Cyprus provides a great example that shows how early farming communities actively balanced local resources, cultural choice, and external influences, following a very unique trajectory. So very briefly, here's a table with calibrated date ranges summarizing cultural evolution in Cyprus. Of course, today I will be focusing on the Middle to early Late Bronze Age. From here, we shift to Southwest Cyprus specifically, and to the site of Kissonerga-Skalia. It is located in the modern village of Kissonerga, about 300 meters from the Mediterranean Sea and roughly 8 kilometers north of the modern town of Paphos. Kissonerga-Skalia has been excavated by Dr. Lindy Crew since 2007. Excavations are still going and have established the first stratified continuous occupation sequence from the late Chalcolithic to the late Cypriot Bronze Age in the Paphos district, which fills an important gap in the archaeological record of the western coast. For this talk, Skalia gives us a way to move beyond the question of which plants were present and towards questions of culinary practice, food preparation, and cuisine in Bronze Age Cyprus.
I'm going to focus specifically on Feature 33, a late Cypriot 1A installation interpreted by Crew and Hill in 2012 as a malting kiln used in beer production, as well as for other cooking activities. This interpretation is important because it places the feature within a relatively short-lived industrial complex at the site, dating to the Middle Cypriot III to Late Cypriot 1A transition, which dates to about 1750 to 1550 BCE. For today, I use that interpretation as a starting point before turning to the archaeobotanical and charred food evidence to ask what the plants remains themselves suggest about activity within this feature. This plan shows the broader occupation surface and the location of feature 33 within that activity area. I don't want to go too deep into the architectural details here, but the key point is that feature 33 was not an isolated installation. It was part of a larger working space with associated features, deposits, and material culture. That matters because the interpretation of the charred food remains depends on more than just the microscopic structure. It also depends on where they were recovered, what other materials were associated with them, and what kinds of activities the feature itself could have supported. One reason feature 33 is especially interesting is that it has also been explored through experimental archaeology. Experimental reconstructions by Ian Hill and the HARP team demonstrated that this type of structure could function effectively as a drying kiln. For this talk, that matters because it supports the broader interpretation of feature 33 as a processing installation. It also fits the theme of archaeology in action, bringing together excavation, archaeobotany, experimental reconstruction, and microscopy to understand how food was made and how it was transformed. Since 2007, more than 288 flotation samples have been collected and analyzed from Kissonerga-Skalia. That totals over about 6,500 liters of soil. I previously published the first 92 samples as part of my doctoral research, and later oversaw the analysis of an additional 36 samples for Gregory Thomas' MSc thesis at University College London. The remaining 160 samples have not yet been published. Today, however, I'm focusing only on the material most relevant to this talk, the amorphous charred objects recovered from the 2009 excavation season and the associated archaeobotanical data presented in my PhD. With the recovery process and data set in place, I now want to look more briefly at the broader archaeobotanical pattern before narrowing it down to feature 33 itself. So far, we have recovered and published around 1,927 identifiable plant remains from the site. These include hulled barley, einkorn wheat, emmer wheat, pulses, including chickpea and lentil, and a variety of fruits and nuts, including fig, pomegranate, grape, pistachia, and even flax. So also, we see several edible wild taxa and plants associated with cultivation practices. Essentially, this gives us a broad ingredient base for thinking about food practices at the site. When we look at the current island-wide data, Kissonerga-Skalia is among the top 10 Cypriot sites with charred plant remains. The top 15 sites represent 97.7% of the total identifiable charred remains from the island. Several sites have produced especially large assemblages, including aceramic Neolithic Kyriakatia and Chalcolithic Kissonerga-Maluthia. What is important here is the clustering of productive archaeobotanical sites in southwest Cyprus. In addition to Kissonerga-Skalia, we have Kissonerga-Maluthia, Kissonerga-Masfilia, and nearby Limba. So Skalia is not an isolated data set. It sits within a region that has produced some of the richest archaeobotanical evidence on the island. For the 2007 to 2014 dataset, I first look at the broader archaeobotanical pattern from Kissonerga-Skalia before narrowing it down to feature 33. This gives us a baseline for the kinds of plants present across the excavated areas. The dataset includes 3,402 liters of sediment floated and 1,927 identifiable items. In terms of percentage presence, the most frequent taxa are pistachia, fig, mallow, lentil, heliotrope, ryegrass, some indeterminate grasses, grape, and indeterminate cereals. What is useful here is that Scalia is not dominated by cereal remains in the way we might expect from the mainland Southwest Asian sites of similar age. Instead, the assemblage is relatively diverse, with cereals, pulses, fruits, nuts, and wild herbaceous taxa all present. The next step is to move from this very broad site pattern into the more specific context associated with feature 33. Where the charred food remains allow us to ask a different kind of question, not just what plants were present, but how some of those ingredients may have been transformed into food in an oven context. This chart shows the same 2007 to 2014 Archaeobotanical dataset, but organized by trench density measured as items per liter. What stands out is that Trench B, where feature 33 is located, is one of the areas with the highest archaeobotanical density at the site. Trench D is higher overall, but Trench B is still clearly elevated compared with most other trenches or areas. This matters because feature 3 is not just visually unusual archaeology with the oven, it sits within one of the most archaeobotanically productive areas of the site. So this case study I turn to next is not being pulled from an isolated or random context. It comes from a feature, a trench in an area where plant remains are well represented. From here, I narrow the scale again, from site-wide patterns to Trench B and now to feature 33. So this feature itself shows a more concentrated pattern. When the samples are separated into the core of feature 33, the embedded pithos, and the associated material, the pithos stands out very clearly. It has the highest density of archaeobotanical remains at just over 4 items per liter. That matters because this is not simply more plant material in a general sense. It suggests that the embedded pithos was an especially important depositional context within the feature. So at this point, the question becomes more focused. What exactly is preserved in this core area and within that vessel? And what can it tell us about the activities taking place in and around this feature? Looking specifically at the pithos embedded within the feature, the assemblage is very different from the broader site pattern. The dominant remains are fig seeds with smaller amounts of cereal, grape, and pistacia. I want to be careful here because fig is always tricky archaeobotanically. A single fig fruit can contain many hundreds of small seeds, so a high seed count does not necessarily mean hundreds of individual seeds, or hundred of individual figs, rather. But the concentration is still meaningful. It suggests that fig was present in this pithos context in a way that stands out from the surrounding deposits. For me, this is where the context becomes very important. These remains came from a vessel embedded within the installation, so they give us a more focused view of activity within the feature. To put the plant data from Kissonerga-Skalia into a broader, island-wide context, this slide shows the most numerous plant taxa recovered across Cyprus more generally. The assemblage includes many of the taxa we might expect, including gloom wheats, barley, lentils, chickpea, fig, grape, flax, and pistacia or terebinth. But what is also important is the presence of wild, arable, and weedy taxa, including grasses like lolium, malva, and other herbaceous plants. Here I've highlighted in orange the edible or potentially food-related plants from the Cypriot Archaeobotanical dataset as a whole. Together, these taxa make up about 90% of the identified plant remains from all sites. This is not a marginal pattern. Possible food plants dominate the assemblage. Some of these taxa are clearly cultivated or intentionally used food plants, such as gloom wheats, barley, lentil, chickpea, grape, fig, olive, and pistacia. Others, such as lolium, malva, and other wild or arable taxa could represent weeds or even crop processing byproducts, fodder, fuel, and other possible food resources. So the point is not to say these are definitely the foods people ate. The point is that the Cypriot Archaeobotanical data provide a broad picture of potential food resources or foodstuffs that align with what we see at Kissonerga-Skalia. So if we bring this back to the model, the island-wide archaeobotanical data gives us the possible ingredients. Number 1 on the schematic highlighted in orange. We can see cereals, pulses, fruits, nuts, and other edible or potentially useful plants. But again, these taxa are still only the starting point. They are not yet cuisine. To move from plants to food, we have to ask what happened after collection or harvest. Were these ingredients ground or soaked, roasted, cooked, stored? Were they transformed through ovens, pots, hearths, or maybe other technologies? This is where archaeology and archaeobotany have to work together. The plant remains show us what was available and used, but the context, the installations, the vessels, and charred food remains help us understand how these plants became prepared, culturally meaningful food. So with that context in place, I now turn to the ACOs, or those amorphous charred objects themselves. All of the objects that I'll discuss were recovered through method of flotation, so they are charred, and found alongside fig, cereals, and pistacia within feature 33. Before presenting the ACOs themselves, I want to return briefly to the context from which they were removed. I present 3 fragments, all from feature 33, 2 from unit 233, and 1 from unit 272, which is associated with the Pithos P270. And here we are. These are the ACOs that I will be presenting today. The 2 on the left are from unit 233, and the 1 on the right is from unit 272. All date to the Middle Cypriot III to Late Cypriot 1A transition. I have been working on this material in collaboration with Yuchin Khan at the Institute of Archaeology, University College London, and we are preparing the results for publication. So today what I am presenting is essentially preliminary work. So the first example that I'll discuss is from Unit 233. On the left is what the fragment looks like under binocular microscope. It is tiny, black, irregular, and honestly, at this scale, it would be very easy to classify it simply as amorphous charred material and move on. This is exactly what I did originally in my first analysis of the material in 2009. But when we view the same fragment with SEM, it becomes much more informative. It is not just a random lump. We can see texture, we can see voids, compacted surfaces, and bits of plant material embedded within it. This is where the fragment starts to become evidence for food processing. A boiled preparation, for example, can preserve a very different texture from a baked or a dry cooked one. So these are the kind of details that we're investigating. In the SEM images of this specimen, when we take a closer look, we are able to see fragments of cereal alongside pieces of pistacia. These fragments are surrounded by additional processed plant material that appears to have been altered by heat and fragmented from a larger amalgamation. In the upper right image, we can distinguish cereal tissue or bran fragments, and in the lower images, we can see fragmented pistacia shell. So taken together, the microstructure suggests a processed mixture of cereal and pistacia. These ingredients were not simply present in the same deposit, they appear to have been incorporated into a single charred mass, likely through mixing and heating in or around an oven-like installation. That matters because it shifts the interpretation from identifying taxa to reconstructing a possible food preparation event. The case for food preparation is strengthened by the archaeological context in which the fragment was recovered. So here is an even more detailed image of the cereal bran. What is especially useful about this view is that the cereal and pistachia are not isolated from one another. They are embedded within a broader matrix of processed plant material that appears to have been altered by heat and eventually charred. So this shows similarities with other examples of baked cereal-based or bread-like products analyzed throughout Southwest Asia. So whether we call it bread depends on how broadly we define bread. What we can say more confidently or minimally is that cereals and pistachia were processed together and dry cooked or baked, likely in an oven-like installation. That is based on the context from which they were recovered. To me, minimally, we can suggest a baked cereal-based product, possibly bread-like. To help identify the pistachia, we compared the archaeological fragment with Pistachia terebinthus and others from the modern reference collection at UCL. These images show the mesocarp and the endocarp and the kinds of structures we would expect to recognize under SEM. This comparison matters because the pistachia evidence is not sitting separately as a nutshell fragment. It is embedded within the amorphous charred matrix. The reference collection gives us a way to interpret what we are seeing in the archaeological sample. Here we have the comparison in SEM images. We have the reference material on the left and the archaeological material on the right, and you can see clearly the resemblance between the two. And here's the second example or specimen from Unit 233. As you can see, on the left is the binocular image, and on the right is the SEM. Compared to the first specimen, it shows pretty much the same thing. Pistachia, cereals, charred together with a matrix, and it looks like it was processed or cooked using dry cooking practices. So if we look at this specimen at even higher magnification, we can see that pistachia embedded in that matrix even more. Very similar to the first fragment, perhaps they even came from the same amalgamation. The final example comes from Unit 272, which was again associated with that Pithos 270. At low magnification, it appears another small amorphous fragments. We have the binocular on the left, and the SEM on the right reveals a very different kind of internal structure. Here the close-up shows elongated cellular tissue with measurements marked in the upper right. This likely indicates cereal tissue, but we plan to investigate this specimen in more detail soon. But for now, I include it because it shows the range of structures preserved within the ACOs recovered from Feature 33. Taken together, these examples bring us back to the larger question of what they can tell us about food preparation at Kissonerga-Skalia. Taken together, the ACOs have the potential to provide direct evidence for food preparation. They give us insight to cuisine by understanding the way foods were processed, especially cereals and pistachia at the site, and it also opens questions about culinary choice. Regarding choice, could these charred fragments represent an early form of tremethopita, a bread still made in Cyprus today? Tremethopita is a traditional Cypriot bread made with tremethos or Pistachia terebinthus, the fruit of the terebinth tree. The simple ingredients include flour, water, and terebinth fruits, with some variations incorporating something sweet like raisins. To explore the possibility further, I collected examples of Trematophita from Cypriot friends and from the hills of the Troodos Mountains. Yuchin charred these and we photographed them using the SEM. What we have here are two examples, and within them you can see visible specimens of Pistacia terebinthus embedded in baked modern wheat flour. And of course, the modern wheat would not have been the case for the ethnographic record and prehistorically, but it gives us an idea of what it might look like.
If the connection could be made, I think it would be a fascinating case study that shows 3,000 years of culinary traditions linking Bronze Age to modern day Cypriot cuisine. It's something that I plan to explore soon.
This brings me back to the question I posed earlier. If Cyprus followed a different trajectory from the mainland, from the Neolithic to the Bronze Age, did food culture diverge as well? So I do not think that the answer is a simple yes or no. The evidence from feature 33 at Kissonerga-Skalia does not show isolation from assemblages in other regions of the island or across Southwest Asia. And I think it might be too early to show direct continuity with earlier Neolithic and Chalcolithic assemblages and modern food traditions. I think what it does show for now is a very unique local example of food preparation and technology, including cereals and fruits and pistacia. However, saying that, feature 33 matters because it gives us a rare opportunity to connect several lines of evidence in one place. We are not looking at plant remains, charred amorphous fragments, architecture, and material culture in isolation. We have an installation associated with processing an embedded pithos, Archaeobotanical remains that were charred, ACOs, and experimental evidence suggesting the feature could function as a drying kiln very effectively. So when these lines of evidence are brought together, those ACOs, those charred food fragments, become more than miscellaneous material. They become part of a broader reconstruction of food preparation and consumption practices in Southwest Cyprus, during the Bronze Age, of course.
So returning to the post-harvest intensification model, feature 33 helps us see several stages of the process come together. The plant remains, number one, give us the possible ingredients. The installation and the pithos point to processing and cooking technologies, which is the number 4. The ACOs preserve traces of mixing, heating, and food preparation. So we are not only just looking at the ingredients, gloom wheats, barley, pistachio, pulses, and wild taxa as separate categories, the end-all be-all. We are seeing evidence for plants transformed through labor, heat, and technology into a prepared and culturally meaningful food.
So I want to end by returning to the idea of archaeology in action. These fragments began as small, easily overlooked pieces of carbonized material, but throughout flotation, microscopy, comparison, and archaeological context, they have become evidence for ingredients, processing, cooking, and culinary practice. So at the site of Kissonerga-Skalia, the ACOs help shift that interpretations from what plants were present to what people were doing with them. This is the real contribution here, moving archaeobotanical data to culinary tradition in prehistoric Cyprus. So thank you so much for your time, and I look forward to your questions and to sharing more results as the work develops.
Okay, well, I can start while people
Yeah.
think of questions. So it sounded like the amorphous samples that you're analyzing, it sounds like that's a new methodology, right? And I'm wondering if you know if so we find a lot of materials in Egypt where I work, I don't do any of the analysis part, but I've excavated a lot of it. And I'm wondering if you know if this type of analysis has been applied elsewhere or if this is really something that you're starting
Yeah.
in Cyprus.
I mean, it's a big project. I mean, a lot of people are doing it. So, um, but you have to have the remains. So lots of work has been done at Çatalhöyük. Um, there's been stuff done in Egypt, right?
Yeah.
All throughout Europe. Cyprus, our samples are really quite poor, to be honest. So the
Oh,
fact
okay.
that I was even able to get this, I think these are probably the only ones on the island so far. So now that we know what to look for, maybe we'll get more. But like historically, they haven't really taken a second look. But now we have, there's established protocols, let's say, to analyzing these materials. That's all changing. Yeah. So
Yeah.
lots of
No,
different places, mostly
okay.
a few sites in the Near East. Now I have Yuchin Khan is working in there's some students working in China, um, Japan, Korea, all throughout Europe, and there has been stuff done in Egypt as well. So
That's great. Yeah. Our preservation tends to be really good.
yeah, yeah.
So yeah, a lot of
And I'm actually going to be working on some of that in the next month. So
Oh, great.
yeah, some stuff that's housed at the BM, so it'll be exciting.
From where? Do you
From
know?
Egypt. I don't know the context of
Okay.
it yet, but I'll be doing some SEM stuff the
Great.
next month.
Okay.
Yeah, I know.
Awesome. There is a question in the chat. Do you think the pistachio shell fragments were deliberately included or were they accidentally introduced along with ground nut meats?
Okay, thank you so much, Erin, for asking that question. Because the other slideshow I was meant to share, this was the wrong one. The one that will be uploaded will discuss this. So I presented last year as part of my sabbatical research, or maybe it was 2 years ago. So Pistacia is an interesting species. When I originally found the Pistacia in the samples from during my PhD, I hinted at the likelihood that they could be used for it could be evidence of dung burning is what I used, what I originally thought, and there had been some publications that hinted at this. And then when I saw these charred amorphous fragments, and I saw the cereals and fragments of the pistacia kind of put together in that way, I started doing some research, and I was actually in Cyprus looking at these. And there's a distinctly Cypriot bread product called tremethopita. And tremethos, it means pistacia, or terebinth. It means it's a terebinth in Greek. So there's a long history in the ethnographic record and modern day of pistacia terebinth bread. And it's essentially exactly what we see here. Cereals, ground cereals with the whole or fragmented terebinth seeds. So the reason why I couldn't share the other one is because I had already put up, I connected it with the audio and the video. So that's why I couldn't put it up. But I do talk about it in the other video. An interesting thing about Tremethopita, I went up to the hills in the Troodos Mountains about a year and a half ago, and up by the Mouflon Sanctuary, a lady was selling Tremethopita. And it's kind of like, think of those hard rusks that goat herders will take Tremethopita, which is bread with this pistacia in it, and they'll dip it in water and they'll soak it up. So it's very hard rust type thing, right? So I kind of went down that rabbit hole, and it is a food that is still eaten in Cyprus today, Tremethopita. So this bread with pistacia is a known food, and it's part of traditional cuisine. And there's evidence in the ethnographic record as well, which on the other video I talk about some work that had been done around 1910, 1915. It was documented. And the interesting thing about that, which I'm writing up, is it's part of a bigger project. So cereals in prehistoric Cyprus are really rare, and they're not very they don't appear like they do everywhere else. Very low quantities. But there are some sites, in particular Southwest Cyprus, during the Chalcolithic, that has large quantities of lolium, which is ryegrass. And you can see on here massive quantities of ryegrass, particularly in Southwest Cyprus. Also where I got that Tremethopita in the foothills of the Trotos. So I think there is room to argue for continued cultural tradition from the Bronze Age when we have the same types of ingredients in that region of the island. It's quite fascinating. It's something that I'm going to explore further. And lolium is getting more traction because more and more people are now questioning, was it a food resource? And I mean, one can argue that it has been used intentionally as a food resource in the past. So yeah, so thank you for asking that. I can go on and on about that because it's a fascinating. So I just didn't have time to talk about it here. So it was like, I want to hint at it, but yeah.
That's fascinating, the connection between, if it is a connection between the Bronze Age food and what they're still eating.
There's so much culturally in history has gone on since then. It's long removed. But the fact that they're still using the terebinth is, it says something about the environment and the resources. Yeah.
Absolutely. Rebecca?
Hi. So thank you so much for your talk. It was fun to join. I recently started collaborating with Dr. Crew to do residue analysis on some of the molecular scale residue analysis. So, which I have not yet gotten to, but that's this summer's project. But I'm really, really excited by this, partly just because food in general. And I love your kind of like, let's go from just identifying stuff to what are people actually doing? Because that's what's more fun.
Yeah, definitely.
So I guess I would wonder how do you interpret the ACOs and these two particular features within the context of the other archaeobotanical materials that were also found within the Pithos and within feature 233? Can we start to talk about them in association with each other?
Yeah.
What do we think that that's going to say then about practices going on?
Yeah. So I think let me go back to that slide just so that I can see what I presented here. So in that feature, there wasn't a lot of material. I mean, when we talk about archaeobotanics in Cyprus, we're always talking very low numbers. But what was in the feature was, where did I think it was just, fig seeds, problematic, but we can say fig is there. We have 7 cereal fragments that are not within the charred food remains, a grape and 1 whole pistachio. So there is things in this oven feature. But one of the things I didn't really have time to discuss is the context, right? So this is an oven.
Right.
So that kind of it gives us more credence to talk about these charred food remains, right? If these were just on the floors of a house, right? But these are within the oven feature. And the pot, that Pithos isn't being used as a pot, I should say. It's reused pottery to serve as part of the oven feature for the drying kiln. So originally, before I looked at the charred food remains, I thought that maybe it was dung burning and we didn't have a lot of charcoal remains in it. So I thought maybe it is dung. But then when I found these and I understood possibly what they were doing with the oven feature in terms of drying, and if they were using it for beer production, that could be why we see figs. And I do have other, which I didn't report here, I have other specimens of pieces of fig as well as the fig seeds, but I didn't have time to go into that here within that feature. So they could be using it to dry. They could also, if we want to take it a step further, possibly, they could have been using barley and the fig to create a starter for beer in beer production. That's why I'm kind of exploring the cellular patterns on some of the cereal brand. So there's lots of different things we can say, but because it was recovered in the oven, I think that gives us a little hope, right? Yeah.
Yeah, that's great. I'm really looking forward to seeing what comes out of the students' work and to talking further once hopefully if hopefully we get some degree of preservation
Right, so.
on the molecular scale. So thank you.
I was muted. There was a question that was sent to me, but I asked and they actually want to ask you. So the question is, how do you think AI might affect this analysis, I guess, when you're kind of comparing samples and looking at those measurements?
Interesting. What do you mean AI and developing AI assisted identification of these things?
I think so. That's how I interpreted the
Oh,
question.
I like this. I like that. So I think there's a lot of potential there actually, because then you're kind of taking the bias out, the
Right.
human bias. But obviously, I don't think it could be the only way, but maybe in terms of measurement and stuff. Yeah, I'm not quite sure. I haven't heard of anybody trying it. But I'm curious if I put one, anyways, maybe I'll put a picture of it into AI and see what it tells me. I doubt it will tell me much, but
Yeah, I mean, I think AI has different biases, right? But sometimes it helps with kind of initial identification.
yeah, maybe. Yeah. I saw a question here, but the references for the first couple slides. Yeah, I'll put my email on here if you want to contact me for that. There we go. Yeah.
And the presentation will also be posted on the ASOR website, so you'll be able to access it.
Oh yeah. Okay.
Okay, any other questions? I have one last quick question, more for any students in the room or students who will watch this later on the website, which is our first talk this spring was talking about archaeological science and kind of how to publish archaeological scientific analyses. And for your work, which obviously, as you highlighted many times in this talk, it falls between very, very science heavy stuff, but also you need the rest of the archaeology to make sense of what you're talking about. So I guess I'm wondering the types of venues where you would publish these analyses. Is it kind of really heavy science focused publications or more broader archaeological?
A lot of this stuff lands well in vegetation history and archaeobotany. Journal of Archaeological Science. I think if you take a more anthropological view of it, you could publish in Journal of World Prehistory, because they tend not to want the super nitty-gritty of this stuff. But if you make it a larger picture, which it definitely falls into that. So those are the three that I kind of look at mainly. But vegetation history and archaeobotany is the archaeobotany one for archaeobotanical sciences. Yeah.
Yeah, no, that's great. And for students, again, you're able to kind of scale the research to send it to different places. So Awesome. Okay, it looks like we're done with questions, but this was so great. Thank you again.
Oh, thank you for having me. And